Thank you for the invitation because when I first got the invitation, I was a bit shocked why an organic chemist like me, why I'm invited into this conference. But I think last 2 days or last yesterday and today, I was intrigued by all the talks and it was a fantastic experience. But anyway, so as I said that I'm a organic chemist by training, and my group in IIT Kharagpur are actually looking at some of the phase separation behavior, not to just about the sake of seeing the phase separation, but also to use that to make different kind of bottom-up approaches to make materials. I'm gonna talk about quickly what we actually and where part of chemistry and science we belong. So normally our group actually looking into dynamic adaptive materials, and these dynamic adaptive materials, they are slightly different than the bio-inspired materials. And bio-inspired materials are normally, or as you can see, that hydrophobic surfaces that you can construct by getting inspiration from biology or biological materials, but in that case, they are merely static in sense. Okay, but on the other hand, if you see that the other side of the spectrum, what we see actually living materials. Living materials mean we actually try to mimic biology in a sense of try to make microtubules, which is a fascinating self-assembly process which goes through the assembly of proteins in an out of equilibrium system, which is also fuel driven. And that's what I think we got very excited, where we can actually mimic them from something which you can make in the lab. Also something like division, cell division, or having that's some change in the macroscopic level when in embryo develops. So the whole idea is that we move from the static materials to life-like materials or adaptive materials. They are mostly energy dissipation, active regulatory networks, and system integrations are integrated into the material. So that is something that we are excited about to make different kind of bottom-up approach materials. So that's why our motto mostly goes to count, organize, move, and adapt. So when we talk about life-inspiring materials, the most important or the first thing that we want to look at is a living cell, and living cell is an inspiration for any chemist to actually to understand, or not to understand, not even small miniature version of this cell with a long process that can actually takes a biochemical or chemical signals and gives a proper function in autonomous way. That would be fantastic to even think of. So as we call it, sensor, processor, actor pathways. So most of the cases, this signaling network is very complicated in cells, but in our lab, we try to understand whether we can actually form something which is much more miniature, much more simple. So metabolic soft matter system. Anyway, so this is not something that a lot of people are doing. So it's not only us. Many people have been trying to look at compartments from a very primitive way. Phospholipids, blockopolymers, as well as some fancy molecules like protein polymer conjugates. These actually forms membranes and kind of enclosed a aqueous compartment, and then you can actually put your stuff and you need to see what happens inside. And there's definitely you need to have a transport phenomenon from the external environment. But anyway, so most of the cases, this is a liquid phase separation conference. So one thing that I would like to point out that living cells are not aqueous compartment, they are filled with macromolecules. So they are more like a crowded soup of molecules, and they are bumping into each other. So in a very interestingly, as Janilok said in this paper, there is order in the chaos. So it's kind of fascinating that even in a very macrometer size pocket of a micro reactor, you can think of how things are actually working. But anyway, so As a chemist, we are interested in looking into molecules. One of the molecules in our lab that we work on is DNA, DNA double-strand helix structure. So this is very nice. But also, we are more interested in single-stranded DNA because these are actually more like a flexible polymer with negatively charged in their backbone, and we want to see whether they have interactions with the divalent cations, and then is it possible to even synthesize a crowded confinement that we have been talking about. So it goes back to my postdoc time, 2000, it's almost 6 years ago. We accidentally found something. So we're working on making hydrogel with DNA polymers, but suddenly because of some change in the procedure, we found that this poly A, so poly A is like homopolymer of A, and we heated up this solution in presence of magnesium, and then we found that there is a phase separation at high temperature. So that is like a more like a LCST behavior, it's like polynipon kind of, and this only happens. So suppose if you see it here, 400, you can see the turbidity increases in the solution, and this is actually got us quite intrigued because this is also reversible. So you cool down, your solution becomes clear again. So that means the globular form of the poly A goes to solution. But this doesn't happen in the case of poly G. It kind of phase separates in the same way, but they're all purine bases, so aromatic, larger aromatic surfaces, but they actually get stuck in this globule form when at a high temperature, but doesn't come back. But this kind of behavior is not seen in poly T and poly C. This is quite interesting because it gives us an understanding that why this purine-based polymer kind of gives us this phase separation behavior at high temperature. So of course, this depends on the molecular weight of the polymer. So once you increase the polymer weight, the LCST temperature, there's a sharp point where flexible change goes into globule form, and that actually goes down with the molecular weight. And also if you increase the calcium and magnesium concentration, you see at certain level the temperature goes down and it gets a plateau. It's quite interesting to see that there's some kind of electrostatic interactions and then this kind of charge neutralization. But LCST is quite difficult even to understand from the Fleury-Hagens theory. Anyway, so this is something that we did under the microscope. We hit the solution, and this is a poly A solution, and then we see like a popcorn kind of mechanism, so nucleation growth, and there's also a liquid-like behavior. I don't want to stress that, but it's quite interesting. So now the question is that we wanted to make this crowded environment, but then we got a phase separation in poly A, but then these are reversible. So of course we need to somehow arrest them, right? To kind of really get into this compartment that we have been talking about. So what we try to do here is called like we try to combine these two phenomena. One is the polymer properties of poly A, which is the LCST behavior, and then you have the supramolecular interactions, which is more like a duplex formation. This is DNA. So what we have done, interestingly, we took two polymers, and this is actually a bit different than homopolymers because they have repeating units, and these are actually can be synthesized by RCS, or rolling circle amplification, from a template, circular template, and we synthesize two polymers where you have repeating unit A20 and M. M is just a barcode you can think of. It's a sequence that we just amplified from the circular template. But same way, we synthesize a T20N polymer, which is orthogonal. This sequence, blue one orthogonal to orange, and green one is A20, red one is T20. So when you mix them together, of course, we see A20, T20 interactions in solution. But what we have done, you kind of quickly heat it up to 95 degrees. Now we're talking about two temperatures. The one temperature is the cloud point temperature of the poly A, which is the green polymer, and that we know from the last slides. So we added magnesium 50 millimolar, and then this one doesn't phase separate. We know that. But there's also a temperature called A20, T20 duplex formation, which is slightly higher, the melting temperature. than the TCP. So when we cool down, we go to the high temperature, we kind of see this the globule formation of the green polymer, and red polymer stays in the solution, and we quickly cools down. What happens that the red polymer wraps around in the droplet through and the circumference of the droplet and the surface, and forming these A20 T20 duplexes, and then it's kind of get arrested, the droplet. So now once you kind of cools down, this droplet wants to go back to solution, but because of the formation of this duplex, you kind of arrest this. So this is more like a kinetically trapping of liquid-liquid phase separation droplets. So once we have that, and you have this core barcode and the shell barcode, that is actually the identical of M and N respectively, and we can actually use a die which has this N star and N star, and these dies can actually go and orthogonally go attach there. So the green die goes to the core of the particles, and the red dye goes to the surface of the particle. So this is very interesting because you have a shell and you have a core which is full of liquid DNA, and of course you can see the higher the temperature you go, the structures are getting more defined. So we do FRAP, and then we see that the inside of the particles, they are quite liquid, so if you bleach half of it, but the other side, the shell is more like a hydrogel, so they're very cross-linked. One more thing that we noticed that once we have the only single standard domain, so the poly A, and then if we increase the duplexes inside by just hybridizing the N with the N star, that the polymer, the whole particle swells. And this is because the persistent length of the duplex is longer than the single standard. So, so what happens that it kind of gets very difficult for the droplet to stay in the phase separated form. So it gets much more liquid. But anyway, we did a lot of interesting structure formation by just playing with the LCST of the polymer and also the hybridization of the DNA strands. So this is quite interesting where we have gold nanoparticles, and then we can actually use that to pop the shell by using a 561 laser. So I quickly, this is even a bit old, but still I want to show you that as a chemist we wanted to understand whether this kind of structures or this kind of microscopic particles can be used to do chemical reactions inside. And then if the enzymatic activity in the we can call it intraprotocellular environment, if we call them protocells. So that's how the activity of the enzymes get affected by the crowded environment of the DNA. So this is something we were really interested, but at the end, what we wanted to do, chemical signal get processed, and then we have some kind of downstream signal propagation, either like a tissue kind of structure formation or functional adaptation. So one quick example, this is a catalyst that we synthesize. This is called Grubbs catalyst. It's do reactions which close the rings and release this fluorophore. So you kind of think of as a pro-fluorophore, and this is actually sitting inside a streptavidin pocket because there's a biotin attached to the ruthenium complex, which is the catalyst, and then we can actually change the mutate the enzyme to see whether the activity of the enzyme gets changed in the crowded environment, and we kind of call this as a directed evolution. But anyway, not so important in this talk, but what we wanted to see that the DNA environment has an effect on this artificial metal enzyme. And we have seen that some of the mutation, even though it's very difficult to rationalize those results, but in the screening process that we have done almost 80 proteins, and then we have seen some of them almost increase 5 times activity. So we have also seen that the specific reactions kind of generate a strain in the particles, and also there is a fusion effect that kind of change the structure of the particles. Also there is a interdigitation of the membrane as well as fusion. And one of the other examples where we have a DNA zyme which kind of catalyze the breaking of a RNA bonds in DNA. So that's actually also was used inside because the enzymes can be easily incorporated by using these barcode interactions just to and then you can wash these particles. So there is no excess catalyst floating in the solution. So here also we have seen that the specific quenched substrate can go inside and the catalysis can happen and you see a signal which is changing over time. There's also a certain change. So like once we do the cleaving of a specific substrate and then if it dissolves, that substrate can also comes back to the surface and change the color as well as expose a palindromic sequence which kind of cross-link the particles. So this here we are actually doing a metabolic reaction, catalytic reactions, and then because of the reaction, the particles changes its color as well as try to find each other by duplex-duplex interactions. And this is quite interesting because it shows how a completely inanimate object can actually gives you a structuration where you just add a chemical signals and then the particles kind of change itself and then try to find each other using a tissue kind of structures. But anyway, so very quickly I'll show one more important, so this is not published and this is actually we're trying to do in my group. What we're trying to understand whether since we have a DNA a crowded environment, which is more like a liquid like DNA, and then we can also change the mechanical properties because we can also cross-link the particles, the core of the particles by using DNA, different kind of DNA tools, but also we want to see whether any kind of phase separation of the proteins, which is actually known like a fast protein. This is actually a disordered protein and we can actually want to know whether this kind of proteins can be phase separated and inside, and we can actually see whether the structuration and also various properties can be tuned by the DNA interior. And this is something we are working on. This is a sticker, a special model, but the sticker is light responsive. So every time you switch on a light, you can actually go for a back and forth with different phase separated and liquid particles. Okay, so fast, I think I don't want to go into it because this is very well known. Fast people have been working on this. So the fast actually gets phase separated in this paper, Dorothea Dorman, and she showed that if we change or mutate this NLS region, the fast get phase separated at the cytoplasm region, so that in the nucleus normally fast is well soluble, and this is working. The fast has a lot of activity and functions for the DNA splicing and different kind of factors. And once you have these kind of things, you also see ALS and neurodegenerative diseases. So the fast droplets, they also found that over time, the fast droplets kind of go into solidified switch side of percolation. So like liquid goes to more like a solid like structure over time. So anyway, Johan and Dorothy, so they have been working on this for a long time. So I asked Dorothy that whether they can give me some of their protein. So the protein also have an MDP tag and a GFP tag. And that, of course, it's very interesting that we can actually follow the phase separation. And as you can see from their data in solution, once they have a TEV protease, which is cleave the MDP tag and the phase the fast phase separates. So what we wanted to do to bring the fast protein inside our particles, and because of that, so we have a very high concentrated protein solution which is mixed with DNA. And now, once we have the TEV protease activity kind of get kicked in, and that's why we kind of see whether fast will show any kind of phase separation in the presence of crowded environment of DNA. So that's what we have seen. Quite interestingly, we see that a droplet-like structure formation, and fascinatingly, these structures are quite stable. And we have not seen many fusion, so they are kind of stabilized in this concentration of protein. And so they definitely want to see whether we can actually change the concentration of the protein and we can actually achieve different kind of structuration inside the particles. And once we have done that, so this is actually fast GFP divided by the repeating unit concentration of the polymer. So because we cannot change the concentration of the DNA in the particles too much. So and that is why we kind of try to see whether the ratio changes, and we can actually see from the droplet-like structure. So these are all taken within 1 hour of the experiment. So we didn't age this sample. So all these things you can see that the droplets also it can go slightly spinodal region, but here also once these domains kind of fuse together and then wanted to go to the membrane and weight the membrane and forms these vacuole-like structures, and at some point of time you will not be able to see any phase separation because those exceeding the loading capacity of the particles. We also see that the DNA, which are inside present in the particles, and if we color them, so during the RCA, when we make the polymer, we just covalently attach a dye and the red dye, we see that the DNA and the color of the fast is actually co-localizing. So that means DNA is also getting pulled into the fast droplets, which is quite interesting to even think of. Also, as you can see, we have done some of the aging experiment, and this concentration where we started to see some kind of spinodal droplet, and then we see the weighting of the droplet or the membrane over time once we don't have any duplex domains inside the particles. This is M, this is O20, and once we have a duplex domain, this kind of phase separation is bit changing because the duplex, the double stranded DNA, they don't really like, Fas don't like them. So they don't want to get incorporated inside the particles because of the problem with their phase separation behavior. So it's more like antagonistic behavior. And then you see there are a lot of droplets are forming outside the particles. And once you kind of see there's a RNA, which is known from the previous studies, that RNA, particular sequence of RNA is called SON RNA, and that is actually binds to Fas even stronger. So even if you have duplex domains, you can increase the RNA sequence inside, install it, and you can also see the phase separation again increases and propensity of the phase separation increases. So once we did this kind of mobility assay, we have seen that A20 is the very bad binder for the Fas, but M has a quite high binding affinity, but also M star RNA plus, which is definitely the domain of the barcode that we already have, the complementary of it, and the RNA plus, which is known already, they have a very high binding with the Fas. And that is also we have seen, we increase the concentration of RNA, we also see over time the soluble part of the so it is kind of early phase separation by just introducing the RNA, even though there's a 22 place has a very low affinity towards the Fas. Yeah, so one more thing that I wanted to show you that if we cross-link the DNA inside, so now we are changing the mechanical property of the code, and that actually has a huge effect because now the Fas is actually, this is Fas MVP before we added TEV, and once we started doing this, this is 0%, so you have seen already the phase separation behavior, but once we increase the cross-linking, we kind of suppress the phase separation behavior. Yeah. So this is something quite interesting because as the mechanical property changing, the code is less liquid. Now the fast phase separation is suppressed, which kind of brings us to one very interesting experiment. Once we have the cross-link particle, there's no phase separation. After 4 days, old sample, we added RNA and we started to see the phase separation outside the particle. So the fast is kind of moving from, which is a disfavorable situation, from the one where they can bind the RNA outside. So same as well, if we increase the duplex concentration in the shell, normally they like the single strand DNA and they kind of populate in the surface, but once we increase the duplex DNA concentration at the surface, it goes homogeneous. And one more important thing that we have done, this is actually the particles that we A20 and M, there's no duplex domains, and then once we phase separate them with fast, and then if we swell them, we remember we increase the duplex domain inside, the particle swells, and that's why, but because this is already aging, so the protein droplet which is formed, which is kind of weighted the surface, now that kind of solidifies, and then once you add DNA zyme, we kind of get this fast capsule-like structure. So anyway, so I think this is where I'm going to stop because I don't want to, I think we're also over the time. something that I wanted to show, but I think because of the time I could not. So this is something that I talked about, how we can change the spacer sticker model by using a light. So once you increase the concentration of the green the small molecule, which kind of use as a sticker, but the red one doesn't. And we can also see that we can change the phase separation behavior of the DNA inside by using the particular light. We can also go the spinal decomposition, which kind of also goes to the wall, internal wall, and wet the particles. And then depending on the magnesium concentration, this can also have a different structuration. So when through cross-link, we can get a network-like structures where this is kind of doesn't go to the wall, but we can actually go back and form because of the light changing. But anyway, so I think I will take these two questions because I think there's some confusion. Please ask me. So what we're trying to do, I think, is actually try to make microparticles which looks like a living cell, but we also, it's a very minimalistic model to understand liquid-liquid phase separation in a crowded environment in a very controlled fashion. So if you have any questions, please let me know. I'll be very happy to answer. Yeah, so this is my group in IIT Kharagpur. Johan has done a lot of the protein synthesis in mines. Yeah, thank you very much.
Thank you. Thank you, Avik, for finishing on time. We have full 5 minutes for discussion, so go for it. Yeah.
So, Avik, so you showed some slide where you said, the poly-G does not phase separate, aperture and poly-A does. So I'm wondering if that is because in poly-G you have this possibility of quadrupole.
Yes. So it does phase separate, but it doesn't come back to the solution.
So in poly-A, is it possible that the mechanisms where you are forming some interdigitated stacks between different strands?
But during the phase separation process, yes, yeah. So we haven't pulled it because we wanted to see some experimental molecular levels to understand how this is forming. But definitely there is a stacking formation. So this is aromatic.
Yeah, why I'm asking, we did some simulations of poly-A.
Okay.
Where we wanted to look at what is happening at high temperatures.
Yeah.
And what we find, which was surprising, is at low temperature you see this intra-strand stacks sort of breaks, and high temperature it sort of interdifferentiates equal different strands.
Okay.
That could be one of the possibilities.
That could be, but I think still we will not be able to see that in the experimental setup. But there are some recent paper from Priya Banerjee's group, but they are looking at the RNA part of the polymer. So they also see this kind of similar behavior LCST pair.
Yeah.
Yes.
Yeah, so I'm curious about the wetting behavior. So you see wetting inside the structure and also on top, like on the surface of the thing.
So yeah, so if you go back now, so please.
So I'm more interested on the ones that you form on the top, like on the actual surface of the things.
So
you actually you see, right, there is wetting on the surface.
Yeah.
So there can you measure the angle and really characterize the
Just a contact angle, yeah. So that is we haven't done yet, but I think this is going to be one of the. But I don't know how valid this would be because this is not like in a three-dimensional and depends on the.
You couldn't do that. I mean, there's been some methods you can still do it, even in curved structure. Curves. But the question is then, what is your tuning now? Like, what can you change the temperature? What are the things that you can control? I mean, because can you change the temperature or so the interaction of these molecules to the surface?
Yeah. So what we have seen that when the fast is kind of loaded into the particles before the phase separation, but they have a lot of duplex domains, so the polymer is much more stiff. And then we have seen that most of the cases when you add RNA, which is kind of sitting at the surface, fast kind of likes to go back and finds their own RNA and kind of phase separate at the surface. So this is the tuning so far we have done, but we haven't done any change of the metal ions and also the temperature, because I think fast phase separation, I'm not really sure how the temperature behavior, because I know there's if you go to low temperature, I think this will the affinity of the fast interaction will increase, but I think we haven't tried this, but I think this is only one of the ways to tune the properties.
I have a request.
Yeah.
I'm an experimental biologist, so I'm sorry if it comes across to you as that why am I asking this? So this looks fantastic. I'm just trying to understand. Your aim is not probably to understand the disease better, right? I mean, FOS is just happens to be a protein that you used in your system. Is that
Yes. So, what the main idea when we started this is that how does the phase separation would happen in a confined state? Like, is this this kind of some kind of structuration that we see in solution will be still accessible, or we'll see, because at the end what it's doing is even the concentration is increasing, right? The local concentration is increasing, but we haven't looked into it. But then the question also always asks that how fast is get solubilized in the nucleus but not in the cytoplasm, even though there's this very small mutation in the FAS. And then our model system is also a very high concentration of DNA concentrate or condensate. So that means there is some kind of resemblance. We can put, even though this model is still very primitive level because it's only A2MT and specific, but if you increase the concentration of genes of the histone proteins, will this be a mimic of a nucleus? And
okay? So you do want to know
yes, that is
of the disease using this as a system now? Is that
I don't want to say that, yes, that is, I'm thinking that this will give us some insight that cannot be done in the living cell because this is going to be a model system.
okay? So that my follow-up question is that nuclear condensates of FAS are also known, right? Which is where at some concentration of DNA and RNA you see it's happening here, right?
Yeah.
So this kind of a confined system, what additional information does it provide as a book? So for example, MBD tagged in vitro sedimentation assays that Dorothy does and other people do are well standardized. Here, what is the new thing that we can learn using this kind of a system?
So far we are looking at the properties of the FAS, not even like changing, like not trying to correlate what we have in the nucleus. Of course, there are many factors in the nucleus that is difficult to recreate in this synthetic model. But what our goal is to kind of see that the mechanical properties and the crowding all and the hydrodynamic confinement, how we can actually slowly understand that even this is a model protein, but how we can actually understand the phase separation behavior, as well as the idea would be to actually go into more active proteins. So if we can do a reactions which kind of form in situ phase separation, is this reactions gonna be enhanced once we have the droplet inside these particles?
So you might want to take mutants and
Yes.
test that and see how they
Yeah.
All right, wonderful, fantastic. Yes, please.
Fascinating work. So I have questions like you showed spinodal decomposition as well. So how do you characterize spinodal decomposition as compared to binodal? Because it has a very different property, like it is a nucleation limited phenomena. So how do you characterize that? Like it's based on like structure, morphology we're observing, or there is other way to characterize it?
Yes. So I think so the nucleation and the growth process is actually a bit different than once we see in the confinement, because the timeline is different. Sometimes it get very delayed because of the diffusion coefficient of the molecules that's different in the confinement. So come to your questions. Yes, we didn't do any simulation, or couldn't do any phase diagram, because the concentration of the DNA inside the particles is very difficult to change for us. We can alter some, but it's very difficult to change in a very systematic way. But we are looking at the particles from the microscope, and we're trying to understand the concentration of protein where actually this specific droplet goes to the spinodal decomposition.
So, like, how to characterize a spinodal decomposition? Like, from the imaging, how do you know that this is a spinodal decomposition as compared to it's not like phase separation going like?
So you mean saying that the because spinodal decomposition, so you have the metastable structure.
Like,
Yeah, so you have the two phase of the diagram. So you have a, if you see that there is a and there's a little bit of fluctuation kind of change the goes to the spinodal. So the spinodal, you can see the concept of change in the microscope. So you have concentration increasing, you have but the spinodal is also, because of the concentration dependent, also you might not see that that the droplet forms and then droplet goes fuse and go to the spinal nerve, that we don't see it, but we can directly jump to the spinal decomposition because that is the concentration that approves that. So it's it's a more visualizing the kinetics of it, I hope I answered the question.
All right. Let's thank Avik for a wonderful talk. And we now move on to the next speaker, that is Snigdha Thakur from ICET Bhopal. She will talk to us about dynamics of flexible active polymers.